Good evening, students. Uh, Today we will discuss embryo development. So, just give me 5 minutes. What about others like Kalai, Durga, Padmavati? What about others, Sama? Just you inform your friends because only 3 members is not good. So just you join the class immediately. Just inform to your friends. Okay.
Okay, sir.
Okay, sir.
Hello. What about others? Only 5 members are there. Kala. Kala. Hello.
Yes,
Sir.
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 Dasma. This strength are there. And one, four, five.
Sir.
Hello.
They will join slowly, I think, sir.
Sir, okay. I will start. Give me a minute. Okay.
Yes, sir.
So today we will discuss. Dear students, my voice is audible.
Yes, sir.
Yes, sir.
Yes. So today we will discuss embryo development. So in the what is the previous class we done? Endosperm development. What is the endosperm? What is the endosperm? Endosperm is the
Nutritive tissue.
nutritive tissue for the developing embryo in the phanerograms, in gymnosperms. What is the nature of gymnosperm, endosperm? Haploid condition. So it is the endosperm in the gymnosperm formed before the fertilization, right? So in angiosperm, the nature of endosperm is the triploid. Triploid means 3n number, developed from the primary endosperm nucleus. It is after the fertilization only. So these are the regarding endosperms. So it is already in the last class we discussed in the development stages of the endosperms and the types of the endosperms. How many types of endosperms?
3 types.
Nuclear endosperm, cellular endosperm,
Hello, dear.
halobial endosperm, right? And also in every like endosperm, it is haustoria. It is consist of haustoria, right? So and also the another type is ruminate endosperm, right? And also another one is the alluron layer. Okay, these are the last class we completed. So today, uh, embryo development. What is the embryo? Yes, the embryo is it is fertilized egg is called zygote. Fertilized egg, it is called zygote. Yes, my voice is audible. What is the embryo?
Fertilized egg is
Fertilized
called.
egg.
Okay. So what is the fertilized egg? Before formation of zygote, what happening?
Fusion of egg cell with one male gamete and another male gamete with the secondary nuclei. So the fusion of one male gamete and the egg cell is called as.
What about female? The fusion of gametes. What happening next?
Fusion of male and female gametes.
Okay. Fusion of male and female gametes. Next, what happening? The formation of
Formation of zygote is
Zygote.
taking place.
yes.
The formation of zygote after many divisions is embryos.
So here the fertilized egg. Fertilized means before that the fusion of gametes is done. The before that so many divisions are completed. Then the fertilized egg, what we call zygote, right? Zygote. In the mother cell of the sporophytic generation, following a number of predetermined divisions, the zygote gives rise to an embryo. Number of divisions. Number of divisions. Either may be mitotic division, either may be meiotic division, whatever may be. Okay. So do you understand?
Yes,
Number
sir.
of
Yes, sir.
divisions. Simply what we call simply the number of divisions. The zygote, it is convert or an embryo, right? Clear? The development of embryo from the zygote is called embryogenesis. What is this? What is the embryogenesis? The development of embryo from
From zygote.
yes, tell me. Development of embryo from yes, tell me. The development of embryo from the Where is it? The
Zygote.
embryo. Yes, zygote. The development of embryo from the zygote. This process called as embryogenesis. What we called embryogenesis.
Genesis.
So here, dicotyledon embryo, monocotyledon. The development of embryo from the zygote is called embryogenesis. The dicotyledonous embryo has two cotyledons attached to an embryonic axis. Attached to the embryonic attach. So here, the terminal part of the axis above the level of the cotyledons is known as the epicotyle. What we call epicotyle. So, what is the meaning of epicotyle? Just anyone? Epicotyle.
Above the cotyledons.
Yes, above the cotyledons or the terminal part of the axis above the level of the cotyledons is known as epicotyle, and it eventually forms the plumule. What we called plumule. The part of embryonal axis below the level of cotyledons is called hypocotyle. Yes, tell me, what is the difference between the hypocotyle and epicotyle? These are two terminologies very, very important in the GK competitive exam persons. What is the meaning of epicotyle? Epicotyle. Yes, tell me. Epicotyle. Epicotyle. Yes, just now someone told me answer, na? Epicotyle.
Terminal part is called epicotyle, sir, above the cotyledons. It forms plumule.
Okay. Okay, right. What about the hypocotyle?
Part
Opposite.
below, sir, below the cotyledons
Yes.
is called hypocotyle.
The part of the axis below the level of the cotyledons is called hypocotyle, and it develops into a radicule, right? And what happening in the epicotyle? It forms plumule. Here, the hypocotyle, it develops into radicule. It is like embryonic root. So here, the monocotyledonous embryo bears only one cotyledon on a lateral side. So you observe this diagrams. The median longitudinal section of the first one is the dicot embryo, and the B is the monocot embryo. You find out this one? Yes, these two diagrams are visible.
Yes, sir.
Yes, sir.
So here, what are the important? Once again I repeat. What is the epicotyle? Epicotyle. Just tell me epicotyle. Epicotyle.
The part which is present above the cotyledon is called as epicotyle, which forms the plumule.
Okay, next hypocotyle.
Part below the level of the cotyledons is called as hypocotyle. It forms the radicule.
Okay. So these are the diagrams. The first one A is the dicot embryo, plumule. These are two cotyledons. Just you observe the two cotyledons, and hypocotyle and radicule, and these are a root cap. So here in the monocot embryo, only one cotyledon, single cotyledon, plumule, and radicule. So these are the diagrams of the monocot embryo and dicot embryo. So next, coming to zygote. So what are the zygote? How it's formed? So here, after fertilization or after syngamy, the zygote undergoes a period of rest period. So, which may vary from the few hours to 4 months. The rest period time is few hours to 4 months. So generally the resting period is shorter, where the endosperm is cellular, then it is a nuclear type. It is a nuclear type. So in angiosperms, the zygote is situated toward the micropolar side of the embryo sac. The basal portion of the zygote is attached to the embryo sac wall, whereas apical part project to the central wall. So once you remember to the embryo sac structure, yes, embryo structure, then you will easily identify these structures. The angiosperms, the zygote is situated toward the micropolar side of the embryo sac. The basal portion of the zygote is attached to the embryo sac wall, whereas the apical part projects the central wall. So the volume of zygote decreases to half of the egg in the 8 to 10 hours after the fertilization. The volume of the zygote decreases to half of the egg. In how much of times it is required? It is 8 to 10 hours after the fertilization. The vacuole present in the micropolar end of the egg, gradually it is disappear and the cytoplasm around the nucleus becomes densely homogeneous. Densely homogeneous. What is the meaning of homogeneous? Yes.
Same type of food.
Yes.
Same type of gene.
Same type of. Yes.
Genetic material.
Okay. Right. So in here the zygote, it consists the cell organelles. What are the cell organelles here? Endoplasmic reticulum and ribosomes, plastids and mitochondria cluster around the zygote nucleus. About the zygote nucleus, the zygote elongates along the long axis of the embryo sac. So the egg wall, which was confined only micropolar end, extend to the chalazal end after the fertilization. The unleakage egg, the zygote is not connected to the synergids or central cell through the plasma desmata. This is regarding about the zygote. Next, embryogenesis. The most of the angiosperms the first division of the zygote is transfers. The resulting is small apical cell and large basal cell. The most of the angiosperms, the first division of the zygote is transfers. The resulting is small apical cell and large basal cells. So here the apical cell projects into the embryo sac, whereas the basal cell lies next to the micropyle. In some members of the Lorenthaceae, the first division of the zygote is longitudinal, but some other taxa zygote divides and the oblique wall. It is this one will seen in the triticum. From this two-celled stage till the differentiation of organs, the embryo is called proembryo. Proembryo. So the diversity, the embryogeny dicotyledons and monocotyledons is largely differences into the further divisions of the basal and apical cells. So these are the regarding about embryogenesis. So we will go for next dicotyledons embryo. Dicotyledons embryo. So what is the dicotyledon embryo? So you first of all, the dicotyledon embryo, how to develop? So here the dicotyledons embryo. So most of the dicotyledons, the apical cell of the two-celled pro-embryo, it here two cells pro-embryo divides longitudinally or transversely, and the basal cell may remain undivided. The basal cell means it is the lower side one. So here the basal cell may remain undivided or may further divided transferly, depend on the division of the apical cell. Apical cell, it is the how it where it is located. The apical means in the above. So here the apical cell pro-embryo may be linear to or T-shaped. So on the basis of the cellular configuration, the embryo of the four-cell stage and the part played each of these cells in embryo development, dicotyledons embryo have been classified to the following 5 types. According to the Indian embryologist, Panchanan Maheshwari. The scientist name is the Panchanan Maheshwari in the 1950. So according to him, the dicotyledon embryos as classified into the following 5 types. How many types? Yes. How many types?
5 types, sir.
5 types. In this, the first type is crucifer or ungrad type. First one is the crucifer. Second one is the aster type. So here the first one is the apical cell of the two-celled pro-embryo divides in the longitudinally. It is again 2 types: crucifer, ungrad type, or aster type. The second one is the apical cell of the two-celled pro-embryo divides transversely. The type is Solon type, Caryophyllite type, Chenopodite type. So these are the 5 types of cells are there. So embryo development, how embryo development, just you see the diagram in the left side. This is the zygote, this is the embryo sac, this embryo sac. These are the terminal cell. It is the apex cell. The lower part is the basal cell.
So here generally the diploid zygote divides form a two-celled pro-embryo. From this two-celled stage up to the differentiation of the organs, the embryo is called pro-embryo. The first mitotic division of the zygote is transverse, splitting the fertilized egg into a large basal cell or suspensor cell close to microphyllae and a small terminal cell or embryo cell project to the embryo sac. So these are the embryo development is continuation with the terminal and the basal cells. The pro-embryo is the suspensor. So the basal cell continues to divide transversely, producing a thread-like cells called the suspensor, which anchors to the embryo to this parental tissue. So the suspensor also functions in the transfer nutrients of the embryo from the parent plant. So here, according to the Maheshwari, the Panchanan Maheshwari crucifer type, this basal cell plays in the no part or only minor part in the subsequent development of the embryo. It is example Anonaceae, Brassicaceae, Onagraceae, Pedaliaceae, Ranunculaceae, Scarpulariaceae. So aster type, the basal cell and apical cell both contribute to the development of a embryo. It is Astraceae, Balsamiaceae, Violaceae, Whiteaceae. These are the example. Next, the apical cell of the two-celled pro-embryo divides the transferly. In this, the first type is the Solon type. The basal cell usually forms as the suspensor. Example is the Campanulaceae, Lineaceae, Solanaceae, Piaceae. So, next one is the Caryophyllite type. The basal cell does not divide further and the suspensory it is present in the derived from the apical cell. Example is the Caryophyllaceae, Crasulaceae, Halorogaceae. So, Chenopodite type. The apical and basal cells both contribute to the development of embryo. This Chenopodiaceae and Boringiaceae. The above 5 types embryogeny refer to those plants where the first division of the zygote is transferred, so that the apical cell and basal cell are formed. And after Maheshwari, the Johansson has called pipered type. It includes cases where the first division of the zygote is vertical. Example is the Loranthaceae and Piperaceae. Some modes of embryo development in the dicotyledonous zygote after transfer section, the crucifer type, aster type, Solon type, Caryophyllite type, Chenopodite type. So these are the entire total embryo development cells. These are the finally the formation of the these are the suspensor part, radical part, cotyledon, plumule. It is the cotyledons, hypocotyl, epicotyl, plumule, radical, root cap. So these are the development stages of the suspense. These are the embryo, the stages of the development of the dicot embryo. First one is the zygote or zoospore. The second one is the division of zygote into suspensor to the embryo cells. Second one, the C, formation of the suspensor and embryo contact. This one. Next D is the periclinal divisions of the embryo octans to form outer dermatogen. This one. Next E, globular embryo showing the region of radical procambilic unit, ground meristem and dermatogen. This one. The next F is heart-shaped embryo. This is the heart-shaped embryo. The next one, the mature dicotyledon embryo, and H is the typical dicot embryo. So these are these stages of the development of embryo, dicot embryo. So the fully formed plant embryo below the Below the features are there. The features are the root shoot axis or embryonal axis. It is the meristems, opposite ends. One or two cotyledons or seed leaves are attached to it. The single sheet-like cotyledons in monocot is called scutellum. So the point of the attachment of cotyledons to the embryonal axis is called cotyledonary node. The portion of embryonal axis above the level of cotyledons is epicotyle. You know epicotyle, you know hypocotyle, which terminates with the plumule or stem tip. The cylindrical portion below the level of the cotyledon is hypocotyle that terminates at the lower end in the radicle or root tip. So these are the fully formed plant embryo. Just you observe the root tip, radicle, hypocotyle, and two cotyledons. This is plumule. This is the epicotyle, hypocotyle, axis, seed coat, and the cotyledons. Fully formed plant embryo. The plumule gives rise to the shoot system. The radicle develops into the root system. The root tip is covered with a root cap. Rarely in plants such as the Utricularia, the mature of embryo is spherical without rudiments of the plumule, the radicle, and cotyledons. So a radial pattern of the protoder, ground meristem, it is the ground tissue, and procambium is the vascular tissue, is differentiated in the plumule and radicle. So next, it is the embryo in the seeds. The embryo, how to locate it in the seeds. So here, the developing embryo remains embedded in the growing endosperm. Both embryo and endosperm remain inside the nucellus. The unused nucellus may remain in some mature seeds called perisperm. Based on the presence or absence of the endosperm, in a fully formed seed, there are two types of seeds, non-endospermous or X-albuminous. It is example is pea, groundnut, grams, mustard. And next one is the endospermous albuminous. Example is castor, coconut, cereals, rubber. So this is the structure of, just you observe the diagram, structure of non-endospermous mature seed with mature embryo. So already in the above slide, based on the presence of the some of the seeds present in the endosperms, it is the formation of fully seeds. There are two types of seeds. Without endospermous, it is also called as hexa-albuminous. Example is the pea seeds, groundnut, grams, and mustards. So another one is the endospermous or albuminous seeds. It is castor, coconut, cereals, and the rubber. So these are the example of the diagram. Just you observe the diagram. Structure of a non-endospermous mature seed with mature embryo. So this one is the epicotyle. This is the lower part is the hypocotyle. These two are the cotyledons. These are the radicle. These are the plumule. These are the seed coat. So what is this? The non-endospermous mature seed with mature embryo. The common bean, the fleshy cotyledons of the common garden bean, dicot stored food was observed from the endosperm when the seeds developed. So the another type is structure of an endospermous mature seed with mature embryo. Mature seed with mature embryo. So here it is example the custard bean. The custard bean has membranous cartilaginous that will absorb the food from the endosperm when the seed germinates. These are the seed coat, endosperm. These are the cotyledons. These are the shoot apex, hypocotyle, and radicle. The next structure of the mature monocot seed with scutellum. Structure of the mature monocot seed with scutellum. So here scutellum cotyledon is part of the endosperm. So it is plumule coleoptile. These are the specialized cotyledon to absorb the nutrients from the endosperm. The nutrients already be discussed in the previous. It is endosperm. It is a nutrient tissue. It provides the all the necessary nutrients. So types of embryos in the dicot seeds. The 5 types of embryogeny are reported by the Maheshwari. Already I told you the cellular configuration of embryo at the 4 cell stage, role of each these 4 cells in the organ development. So already I told you crucifer or on-grad type, the asteroid type, solanard type, caryophyllite type. So kinopodite type already we discussed in the previously. So next types of embryogeny. So these are the embryogeny: on-guard, ostrich, solan, and kinopodine, caryophyllite, piperat. These are the types of embryogeny. Just use observe the embryogeny. So typical dicot embryo development. Here, the typical embryo development studied in the Capsella bursa pastrosis. It is the family cruciferous member by Hanstein and Femengene in the 1870. So it is the cruciferous type of development. The first division of the zygote is transfers leading to the formation of basal cell and terminal cell. Basal cell, it is in shortcut TB. In the terminal cell, it is called as the terminal cells. Next, embryo development in the Capsella bursa pastrotis. The typical dicot embryo development. Basal cell divides transferly to form a 2 cells centimeters. And what is the meaning of CI? Okay, so the terminal cell divides longitudinally, resulting in the formation of the reserve T-shaped pro-embryo. It is made up of the 4 cells. So here, each of the 2 terminal cells now divides by the vertical wall, laying at right angle to the first to form the quadrant stage. It is called F. These are the bursa pastoris. These are the typical the 2 terminal cells divide for the vertically right angles to the first division to produce the quadrants. The quadrants cells divide by the transverse cell of the, giving rise to the octant stage. Of this contact, lower 4 cells are called hypobasal or posterior actants, and it is the stem tip and cotyledons and upper for the cells named as the epibasal or anterior actants. So these are the epibasal actants, hypobasal actants. These are the 2. Apical dicot embryo, its continuation, all the 8 cells undergo preclinical divisions, resulting in the 16-celled stage, differentiating in outer dermatogen and inner layer of the cells. The cells of the dermatogen divide anticlinally to give rise to endosperms of embryo. The inner cells by further divisions gives rise to the peribulum or ground meristem is plerome or procambium. The peribulum develops into the cortex and plerome into the stele. So these are the development of stage. So by this time, 2 basal cells and 4 cell pro-embryo divide from a row of 6 to 10 suspensor cells, of which the uppermost cell becomes swollen and vesicular to form a haustorium. The lowermost cell functions as a hypophysis. The cell of hypophysis divide into 2 vertical divisions, right angles to each other to give rise to a 8 cells. The lower 4 these form root cortex initially. The upper 4 from the root cap and root epidermis. Next. So how formation of globular embryo and heart-shaped embryo? The horse-shaped embryo. 3 different types. The first one is the globular embryo. B is the heart-shaped embryo. This one is a horse-shaped embryo. The enlarging the embryo consists of 2 cotyledons and embryonal axis. The hypocotyle as well as cotyledons soon elongate in size. During further development, the oval becomes curved like horseshoes. So here the stages are there 10 to 11. Just wait. This is not required, but they are explaining the some. They are taken in the capsula bursa. So we on our exams we are covered only the general dicotyledon embryo. Development of general characters of dicotyledons of embryo is enough. So they are taken in the slide. Explain the one examples. There is no need of example explain. So next, monocot embryo. Monocot embryo. So here, monocots as a good deal of variation is found in the stages of the development. There is regarding it is the early cell division of the pro-embryo. Early cell division of the pro-embryo. The embryogeny of monocot embryo has been traced out in the typical type Sagittaria, Sagittarifolia and Alasmaceae family. So generally in this 4 stages in the development of the monocot embryo. 4 stages. How many stages? 4 stages. The 4 stages are pro-embryo stage and globular stage and scutular stage and coleoptile stage. These are the 4 stages of the monocot embryo development. So here the first of all zygote greatly enlarges in the size. Here simply the step is the zygote is enlarges in the largely in the size. The first division of the zygote is an asymmetrically transfers, leading to the formation of a large basal cell and small terminal cell. What happening here? The formation of large basal cells and small terminal cell, right? Here also two cells. First one is the large basal cell and second one is the small terminal cell. So larger basal cell which lies towards the micropolar end does not divide the further. Here the micropolar ends does not divide in the further. So it is enlarged and transformed directly to form a large suspensor or vesicular cell. The terminal cell divides transferally and form a tree called proembryo. In the next stage, the lowermost cell of proembryo divides by two vertical divisions, forming a quadrant stage. The quadrant now divide transversely, forming the octants, the 8 cells bearing arranged in the two tiers of 4 cells each. Two tiers of 4 cells of each. So the octants, the results of periclinal division, the octants with the results of periclinal divisions produce the 8 outer and 8 inner cells. How many? 8 outer cells and 8 inner cells. The outer cells develops into a gametogen and inner cell gives rise to the peribelum and plerome. Peribelum and plerome. So the middle cell of the 3-celled proembryo divides transferally, forming the upper and lower cell. The upper cell undergoes repetitive transfer, forming the upper and lower cell. The upper cell undergoes repeated transfer and vertical divisions, differentiation into 3 to 6 suspensor cells, the radical, plumulae, and hypocotyl. So here, these are the stages of development of the typical monophyte embryo. These are the zygote. The next step is the terminal cell. This is the formation of the lower side is the opposite. Terminal cell, this is the basal cell. It is the vascular cell. The formation of the section C is the formation of the vascular cell. And next D, next E. So here, then some of the divisions occurs. The formation of the F and R, finally the vascular cell. These are the suspensor, these are the radical, these are the cotyledons. So next, the 8 cells of the octant stage after differentiating in the peribelum and plerome and dermatogen divide further and form a single cotyledon. These entire process or these entire parts, the 8 cell of the octant stage after differentiating into peribelum, plerome, and dermatogen divides further and form a single cotyledon. The cotyledon is terminal in position. Plumulae is situated laterally. The depression of the basis of the scutellum embryo axis. So the structure of monocot mature embryo is more complex than dicot embryo. So the monocot likes Colocasia, no suspensor is formed. Agapanthus, the family Liliaceae, 2 cotyledons have been reported. So conversely, the dicots like Ranunculaceae, Ficaria, and some members of Apiaceae, Umbelliferae have single cotyledons. So these are the monocot development, monocot embryo development. Those 4 suspensor, middle cell, terminal cells, the suspensor. So here, just you observe that these are the formation of suspensor. After suspensor, they are produced in the hypocotyl, plumulae, radicel, and cotyledons. Then finally we got the monocot embryo. Here, some of the main difference between the monocot embryo and dicot embryo. So here, only one cartilage attached to the embryonal axis. This is the monocot statement. And next one is, there are two cartilages attached to the embryonal axis. So plumella is lateral, plumella occur distally. So a single cartilage occupies the terminal position. The cartilages occur laterally. The envelope of plumella is called coleoptile. The coleoptile is absent. So coleorhiza is a protective sheath of radicle. The coleorhiza is absent. Single cartilage called scutellum is present. Scutellum is absent. Relatively small suspensor. The suspensor is larger. So just you difference between the monocot and dicot embryo. Just you observe that these two embryos. Though this is the bean seed, it is the dicot. You know, this one is the bean seed. It is the two cartilages. So here the corn seed, it is the monocot. So these entire part is these are the embryo. It is the embryo. These are epicotyle, the hypocotyle, radicle, the cartilages. What happening is the same to same. The pods are there, but what happening here? Two cartilages here, only single cartilage, and it is seed coat. Okay, these are the difference between the monocot embryo and dicot embryo. So I will share the one more. So slide is visible.
Yes, sir.
So already we discussed in the lot, but this is very, very important in your PD exams as well as practical exams also. Okay. Yes. Yes, sir.
Monocot and dicot both, sir.
Ah, yes. So it is in the like slide, the permanent slides already prepared. It is available in our laboratory. Just you in the practical exams, in the suppose the minor question, they without any names, the slides kept on the under the Microsoft. Just you go to the Microsoft, just you see, then observe, then you identify. Oh, this is the monocot embryo. Just this is the dicot embryo. Then identify neatly diagram. Then we will some descriptive. So not there is no development stages of the practical. Just what are the main points of the monocot embryo? The characteristics of the monocot embryo or dicot embryo, it is enough. The both are the very important. So here these are the summary of the embryo development. These are the zygote, the formation of apical cell, formation of the basal cell. It is the actant, the formation of from the basal cell, suspensor and hypophysis. Here proximal and diastal, the hypocotyle basal part of the cartilage stem tip. So here monocots. So here one good different one important part also is there, that is suspensor. That is suspensor. The formation of suspensor is constant feature of in the embryogeny of the angiosperms. The suspensor is ephemeral structure found in the near the radicle of the embryo. It grows much faster than the embryo during the during the early stages of the embryo development, and it is usually attains with the maximum size by the globular stage. In a mature seed, it remains the suspensor may be seen. The suspensor shows a great variation with the regards in the size, shape, the number of the component of the cells. The variation are usually related to the its chief function of supply of food materials to developing a embryo. The suspensor attached to the embryo to the wall of the embryo sac and pushes it deep into the endosperms so that embryo lies and nutritionally favorable environment. So here the Lorenthaceae family. The Lorenthaceae family, the embryo sac grows up to various heights in the style. So the egg is fertilized at the tip of the embryo sac in the style, and the endosperm is formed inside the ovary. In this, the plant suspensor is expansionally long so that it is able to bring embryo down from the style into the endosperms. In some plants, the suspensor cells become greatly enlarged and give rise to the prominent ostoria. They penetrate the cells, endosperms, and sometimes even encroach upon the tissues, the oval surrounding the endosperm. As early as Layard had shown the occurrence of the suspensor ostoria in some Rubiaceae members. Since then, the presence of suspensor ostoria has been reported in the several plants. Extensively developed the suspensor ostoria were observed in the families Archidaceae, Podostemaceae, Tropaceae. So the family Holorhizaceae, suspensor of ostoria appear like synergids. The myriphylum, the member of the family, two-celled proembryo comprises a large basal and much smaller in the terminal cell. The basal cell divides longitudinally to form two daughter cells, which enlarge to some extent to the occupy the entire space of the micropolar part of the embryo sac. The nuclear extremely. So here two diagrams are there. These are the suspensor ostoria. The first one is the dicrea, microphyllum, and sedum. These are the key difference. These are the suspensor cells. These are the embryo. These are the proembryo. These are ostorial branches. So in sedum, it is belongs to the family Crassulaceae. So the apical cell of the two-celled embryo gives rise to the embryo proper, as well as four-celled suspensor. The basal cell enlarge any division. The nucleus of the latter becomes a permanently hypertrophic. The cell grows through the nucellar epidermis, forming a vesicle micropillary. The vesicle develops the tubular branches on either side of the embryo. These ostoral branches penetrate through the integuments up to the raphe. So here, today class regarding about the mainly focus in the dicot embryo and monocot embryo. So in your exams, you must read and suspensor is a short answer, and another one is the monocot embryo. Monocot embryo, they are asking sometime in the short answer. Sometimes they are both the embryos, like monocot embryo and dicot embryo. So you compulsory prepare these two types.
Difference adugtara sir?
Definitely, maybe అడిగే ఛాన్స్ ఉందమ్మా. కాకపోతే ఏంటంటే మీరు రెండు చదువుకుంటే it covers both as long answer as well as short answer, but it's very, very important. Okay.
Okay, sir.
So just you tell me what are the remaining topics. Actually today in the I'm in the outside. There is no book. So just you tell me what are the remaining topics.
The development apomixis is there, polyembryony.
Oh, apomixis is there. Okay.
And parthenocarpy.
Okay.
Experimental embryology, applications of embryology in taxonomy, agriculture and horticulture,
Okay.
Principles and applications of palynology.
Okay. So, okay. So in the unit 4, like unit block 3, development of embryos, endosperm, development of embryo. Then next one is apomixis.
Apomixis.
Next,
Polyembryony.
polyembryony. Okay. The remaining these two, and next new block is there. The new block is
Parthenocarpy.
parthenocarpy. And modern. What is this? The topic is modern
Embryology of angiosperms, sir. 3. Block 3.
Okay.
Total
So,
6 units, sir.
total 6 units. And how many class remaining?
Sir.
The last
4 classes is there. 4, sir.
4,
20, 22nd, 24, 27.
27. The last class is the 27, right?
Yes, sir.
Okay, we will in this time we will compulsory complete our syllabus, right?
Okay, sir.
So today, do you understand this one? How to, first of all, these all are the link with the beginning of the flower, the reproductive parts of the flower. That is the base, okay? And the reproductive structure of the flower, it is consists of stamens, the ovary, ovules. So these all the flowering parts compulsory you learn. Then you differentiate what is the ovary, what is the main parts of the ovary are there, okay? Anther. Then we will go for the next part. It is easily understand what is embryo sac, right? What is endosperm? What is embryo? So these are the very important, the basic principles. Once you get the and also types of ovals. It is also very important. The types of ovals, how the oval structure, where it is the located is chalazal end, micropolar end. Okay, what is the terminal side? What is the apex side? These are the some terminology is very important. Understanding of these type of class. These all are the like polyembryony and apomixis and seed structure, de development of embryo, development of endosperm. These are the each one to just together link. So compulsory you learn about the flowering plants or reproductive parts of the plant. Okay, the life cycle of the flowering plant. So just what are the components, what are the major parts in the plants are there? Just you pluck a flower in the physically, just you observe with the cross check with these parts. Oh, this is the petal, this is the sepal. Okay, what are the function of this one? This is this is the stigma, this is the style, this is the pollen grains. Okay, what is the sporangia? What is the sporangia spores? What is the what is the pollen grains? How the structure? How collect the pollen grains from one plant to other plants? How the insects doing there? Just these are the some basic knowledge you learn about the beginning. So directly you open the book. What is the monocot? What is the dicot? You just anyone simply say the answer. Monocot is the two single cartilage and dicot is the two cartilage. Everyone knows this fact, but how it is developed? How it is developed become a monocot embryo? How it is developed become a dicot embryo? How is the development stages are there? How it is involved? What are the enzymes involved? What are the scientists are involved? What are the process? What are the difference between the monocotyledon development and dicotyledon embryo? So every time you just compare the studies, comparison of the studies, it is good for you. So just you see this one. Just you remember this. So these are the already this one. This one summary is there. This one. Just this type of notes you prepare this type of notes. So this is the zygote. What happening in the embryo development stage? Apical cell, the formation of apical cells and basal cells. Then the from the apical cell, actant. From the actant, divide the proximal tire, distal tire. From the proximal tire, the hypocotyle basal part of the cotyledons. Here, distal tire, the cotyledon stem tip. So here, the basal cells, that is the formation of the suspension cell. It is the hypophysis. It is the root tip and root cap. So here, these are the just wait. Kala, only two members are there, Sujata and Kala. What about
Yes.
remaining? They are the middle. The middle they are skipped.
Yes, sir.
Why? What is the problem? So generally, the class is boring. So
No, sir. It is very good, sir.
so actually,
We are understanding, sir.
so it's very simple. So this is only the process is very simple. So just you find out, you catch the what is the development of monocot embryo, development of dicot embryo. What is the differences? Just the remaining process is the same. So here monocot embryo, development of monocot embryo, development of dicot embryo, they develop the basal cell, terminal cell, apical cell. These are the same. What is the difference? What are the exact difference you catch? Just you prepare your notes this type. So again, there is you want notes. I will send you after once the completion of the syllabus. I
Okay,
will
sir.
send all the notes, but you prepare the notes. Just you listen what you understand the class. Then you some flowchart. This flowchart is very important. Very important because it is easy learning process. It is very good learning process. It is very like we are not forget it. So step by step, a very good process. Otherwise, we like story type reading. They can't not enter to the your brains. So it is immediately gone. Once you close the books, it is gone. But after the class, just you prepare. So nowadays, too much of social media is there. Google source, so many books, so many material, YouTube videos there. But once you listen, after listening one topic, then you prepare your own notes. That is good for you, and it is very good for you. It is as well as not only these exams. That is for the competitive exams also useful.
Yes,
Okay.
sir.
So this is the I share with you because so many students, just many, so second year have what class? Nothing. We are directly read the books. Why need the class? Some most of the people thinking this type, but is that is very bad. So because so every time they don't know some of them. So the the teacher explained, for example, you don't you cannot understand. Just you ask me the sir, what is the process? Just to explain. Actually, you are a MSc student, so we are not elaborate in the every point because your stage according all of you, your teachers and many jobbers are there. You know how to teach, how to reach the student. So you are not. I think and my thinking is you are not a like 10th class, 9th class or degree student. You are the masters. Just oh, some sentence I given the sentence. No, no more explanation because your stage is very high. Just you get it the points. That is the good for the for your so whatever maybe. So according to your time, according to your busy schedule, according to your plans or your own risk. So I will suggest it, but any suggestion regarding any material, any further studies, any research type of in your life in the future studies, please ask me. I will help you of my knowledge.
సార్
Thank you,
ఇంపార్టెంట్
sir.
క్వశ్చన్స్ సార్ ఇంపార్టెంట్ క్వశ్చన్స్ మీరు చెప్పారు కదా సార్ టైప్స్ ఆఫ్ ఓవ్యూల్స్ ఇంపార్టెంట్ అని. తర్వాత
ఒక పని
డైకాట్ అండ్ మోనోకాట్ ఎంబ్రియో అని చెప్పారు సార్.
ఒక పని చేద్దాం, మీకు క్లాస్ లాస్ట్ డే కానీ లేకపోతే ఒక రోజు ఒక 10 minutes జూమ్ మీటింగ్ పెడితే నేను యూనిట్ వైజ్ గా అప్ టు అంటే మళ్ళీ ఫస్ట్ నుంచి ఎథనోబాటనీ నుంచి మెడిసినల్ ప్లాంట్ నుంచి ఏమేమి
Yes, sir.
చదవాలి ఏం చదవద్దని చెప్తా. సో హియర్
Yes, sir.
ఇందులో మోనోకాట్ ఎంబ్రియో అండ్ డైకాట్ ఎంబ్రియో ఇస్ వెరీ వెరీ ఇంపార్టెంట్.
Yes, sir.
ఈ రెండు మట్టుకు అయితే.
Yes, sir.
సో స్ట్రక్చర్ వాటి మోనోకాట్ ఎంబ్రియో ఒక్కోసారి ఏంటంటే డెవలప్మెంట్ స్టేజెస్ అడుగుతాడు, ఒక్కొక్కటిసారి ఏంటంటే జస్ట్ మోనోకాట్ ఎంబ్రియో ఆర్ డైకాట్ ఎంబ్రియో అంటాడు షార్ట్ ఆన్సర్లో. మీరు డయాగ్రామ్ వేసి దాని గురించి 5-6 పాయింట్స్ రాస్తే తప్పిద్ది ద నేచర్ ఎట్లా ఉంది ఏంటి అనేది. లాంగ్ క్వశ్చన్ అడిగితే ఈ స్టెప్స్ రాయాలి. బేసల్ హౌ టు ఫస్ట్ ఆఫ్ ఆల్ బేసల్ స్టెల్స్, టెర్మినల్ స్టెల్స్ ఎట్లా ఏర్పడుతున్నాయి, ఎపికల్ స్టేజ్ ఎట్లా ఏర్పడుతుంది, స్టెప్ బై స్టెప్ డయాగ్రామ్ చేసుకొని మీరు రాయాలి. సో కాబట్టి ఇందులో మీరు స్కిప్ ఏం చేయొద్దు, కంపల్సరీ మోనోకాట్ డైకాట్ కవర్ చేస్తే మీకు ఇటు లాంగ్ ఆన్సర్ కు యూజ్ అవుతాయి, షార్ట్ ఆన్సర్ కూడా యూజ్ అవుతాయి. స్కిప్ చేయదు, ఈ రెండు మట్టుకు మాత్రం స్కిప్ చేయదు, మిగతా లైట్ తీసుకోండి. బట్ ఇందులో వెరీ వెరీ ఇంపార్టెంట్. ఓకే
ఓకే సార్.
ఫ్లవరింగ్ ప్లాంట్ ఇంపార్టెంట్ సార్. అంటే ప్రొడక్టివ్ పార్ట్స్ ఆఫ్ ఫ్లవరింగ్ ప్లాంట్ అన్నారు, అది ఇంపార్టెంట్.
ఏంటమ్మా ఏంటంటే నాకు అర్థం కాదు.
ప్రొడక్టివ్ పార్ట్స్ ఆఫ్ ఫ్లవరింగ్ ప్లాంట్ అన్నారు కదా సార్, అది ఇంపార్టెంట్.
నేను ఏమంటున్నానంటే ఫ్లవరింగ్ ప్లాంట్స్ అనేది జస్ట్ జనరల్ నాలెడ్జ్ కోసం చెప్తాను. అంటే ఇవంతా ఏంటి, ఇప్పుడు ఎండోస్పెర్మ్, డైకాట్ ఎంబ్రియోస్, తర్వాత ఓవుల్స్, ఎంబ్రియోస్ ఇవంతా రిలేటెడ్ టు ద ఫ్లవర్. అంటే బేస్ అనేది అక్కడి నుంచి. అంటే మనకు ఫస్ట్ ఆఫ్ ఆల్ మనం ఫ్లవర్ లో ఏం పార్ట్స్ ఉంటాయి ఏంటో తెలియకపోతే ఇవి నీకు ఏం అర్థం కావంటున్నా. అందుకనే అది ఇంపార్టెంట్ అంటున్నా, నీకు అండర్స్టాండింగ్. నీకు ఫ్లవర్ ని డిస్క్రిప్షన్ చేస్తే దాంట్లో పార్ట్స్ ఏముంటాయి, సెపల్స్ అన్ని వేటినేమంటారు. ఒక ఫ్లవర్ ని తీసుకొచ్చి కట్ చేసి మీ పక్కన పెట్టుకోండి. ఇప్పుడు ఏదైతే ఫ్లవర్ నెట్ లో కొట్టినా సరే, నేను ఇచ్చిన క్లాస్ నోట్స్ అయినా చూసుకుంటే తెలుస్తుంది, ఓ ఇందులో సెపల్స్ ఏవి, డిసెక్షన్ చేయండి. లైక్ హైబిస్కస్ రోజు మందార ఎక్కడైనా దొరుకుతుంది కదా, ఆ ప్లాంట్ తీసుకోండి, ఆ ఫ్లవర్ తీసుకోండి. ఒక్కొక్కటి ఇగో కలర్ఫుల్ ఉన్నాయి, సెపల్సా పెటల్సా, తర్వాత బేస్ కింద గ్రీన్ కలర్ ఉంటాయి అవేంటి, తర్వాత స్టిగ్మా ఎట్లా ఉంది, పోలన్ గ్రెయిన్స్ ఎట్లా ఉన్నాయని ఒక్కొక్క పార్ట్ మీరు డిస్కస్ చేయాలి. డిస్కస్ చేస్తే అంటే ఈజీగా అర్థమవుతుంది. ఒక్కసారి ఆ ఫ్లవరింగ్ ప్లాట్స్ ఓవరీ ఇవంతా అర్థమైతే నెక్స్ట్ ఎంబ్రియో సాకు, తర్వాత ఓవుల్స్, టైప్స్ ఆఫ్ ఓవుల్స్ ఎట్లా ఉన్నాయి అనేది, ఇదంతా ఏంటంటే ఒకదానికి ఒకటి లింక్. సో నువ్వు డైరెక్ట్ అక్కడ ఫ్లవర్ ను మర్చిపోయి నీకు ఫ్లవర్ పార్ట్స్ లో ఉన్నటువంటి రిప్రొడక్టివ్ ఆర్గాన్స్ ఏందో తెలియకుండా డైరెక్ట్ నువ్వు వచ్చేసి ఎంబ్రియో సాకు చదువుతుంటే నీకు అర్థం కాదు. నీకు ఓవుల్ చదవకుండా ఓవుల్ స్ట్రక్చర్ ఎట్లా ఉంది, దాంట్లో మైక్రోపోలర్ ఎండ్ ఎట్లా ఉంది, చలాజల్ ఎండ్ ఎట్లా ఉంది, తర్వాత న్యూక్లియస్ ఎట్లా ఉందో అది తెలవకపోతే నీకు ఎంబ్రియో సాక్ తెలియదు. ఎంబ్రియో సాక్ తెలియకపోతే ఎండోస్పెర్మ్ తెలియదు. ఎండోస్పెర్మ్ తెలియకపోతే నీకు ఏంటి మన ఎంబ్రియో తెలియదు. సో దీనికి ఒకదానికొకటి లింక్ ఉన్నాయి కాబట్టి ఫస్ట్ అసలు జైగోట్ అంటే ఏంటి, జైగోట్ ఎట్లా ఫామ్ అవుతుంది, అసలు గామెట్స్ అంటే ఏంటి, గామెట్స్ ఎక్కడి నుంచి వస్తున్నాయి, గామెట్స్ ఏ ఆర్గన్స్ ప్రొడ్యూస్ చేస్తున్నాయి, మేల్ ఎక్కడి నుంచి వస్తుంది, ఫీమేల్ ఎక్కడి నుంచి వస్తుంది, అట్లా ఈ లింక్ అనేది ఉంటే ఈజీగా అర్థమవుతుంది మనకు. సో అంటే బేసిక్స్ అంటే ఇది ఓన్లీ ఎగ్జామ్స్, నేను ఎగ్జామ్ పర్పస్ అని చెప్పట్లేదు, ఎగ్జామ్స్ కోసం అని చెప్పట్లేదు, నీకు నాలెడ్జ్ కోసం అని చెప్తున్నా. రేపు మీరు టీచ్ చేస్తారు లేకపోతే ఇంకొకటి దానికోసం చెప్తున్నా నేను. ఓకేనా? అవే క్వశ్చన్స్ అడగరు. డిస్క్రైబ్ ద ఫ్లవర్ అని మీరు అదే ఏంటి స్కూల్ లెవెల్ క్వశ్చన్ తీసుకొచ్చి ఎంఎస్సీలో అడగరు. బట్ మనం నేర్చుకోవాలంటున్నా నేను, బేసిక్
నాలెడ్జ్ పర్పస్.
అది ఉంటేనే ఇన్ డెప్త్ వెళ్ళగలుగుతాం. వితౌట్ డెప్త్ ఇప్పుడు ఉన్నది డైరెక్ట్ ఎంబ్రియో సాకు వచ్చి నువ్వు అక్కడ ఏమంటుండు, ఎంబ్రియో సాకు వచ్చి చలాజల్ ఎండ్ అంటుండే, మైక్రోపోలార్ ఎండ్ అంటుండే, ఇది ఏందో తెలియకపోతే నీకు ఎట్లా తెలుస్తుంది అసలు ఎంబ్రియో సాకుల ఈ పార్ట్స్ అనేది, అసలు ఎంబ్రియో సాక్ అనేది ఎక్కడ ఉంటుందని తెలవాలిగా. ఓవులు ఉంటుంది కదా, ఆ ఓవులే తెలియకుండా నీకు ఎంబ్రియో సాక్ గురించి నువ్వేం చేసుకుంటావు, బట్టి పడుతావు, 10 నిమిషాలు ఉంటుంది, తర్వాత గుర్తుండదు కదా. సో అది నేను చెప్పేది.
సార్ మెగాస్పోరెంజియం స్ట్రక్చర్ కూడా ఇంపార్టెంట్ కదా సార్?
డెఫినెట్లీ. ఆ రెండిట్లో లాంగ్ క్వశ్చన్ ఏదైనా అడగచ్చు.
మైక్రో అండ్ మెగా.
రెండు. సో నేనేమంటున్నానంటే మీకు సిలబస్ అంతా అయిపోయినాక నేను చెప్తే ఏవేవి ఛాయిస్ తీసుకోవాలి, ఏవేవి ఛాయిస్ తీసుకోవద్దా నేను అందరికి ఎక్స్ప్లెయిన్ చేస్తాను. ఓకేనా?
ఓకే సార్.
మీరు కంపల్సరీ గుర్తు చేయండి నాకు
ఓకే
ఓకే.
సార్.
ఇంపార్టెంట్ క్వశ్చన్స్, తర్వాత ఏమేం క్వశ్చన్స్ చదవాలి, ఏమేం ఛాయిస్ తీసుకోవాలంటే మీకు బటన్ తగ్గేయడానికి నేను చెప్తా. క్వశ్చన్ పేపర్ ఎట్లా ఉంటుంది అంటే ఏ యూనిట్లోకి వెళ్ళి ఎన్ని మార్క్స్ క్వశ్చన్స్ అడుగుతారు, ప్రతి యూనిట్ ఉంది. ప్రతి
Yes, sir.
యూనిట్లోకి వెళ్ళి 2 లాంగ్ క్వశ్చన్స్, 2 షార్ట్ ఆన్సర్స్ అట్లా ఉన్నాయి అనుకో. అప్పుడు మనం ఏం స్కిప్ చేయొచ్చు. గన్ షార్ట్ గా అడిగే క్వశ్చన్స్ ఎవ్రీ ఇయర్ ఏమున్నాయి అట్లా
Yes, sir.
చేస్తే ఎంతో కొంత మనకు
చెప్పండి
కొద్దిగా
సార్.
మీరు ప్రెషర్ తగ్గుతుంది అనమాట.
Yes, sir.
అవును సార్.
ఎందుకంటే మీకున్న తక్కువ షార్ట్ టైంలో ఇక లేకపోతే ఇదంతా చదువుకుంటూ పోవాలంటే మీతో కాదు. ఓకేనా?
Yes, sir.
టైం ఉండదు, మళ్ళీ ఎగ్జామ్స్ గ్యాప్ ఉండదు.
అవును సార్.
Yes, sir.
అదొక పెద్ద ప్రాబ్లం అవుతుంది వాళ్ళకి. ఎన్నిసార్లు చెప్పినా కూడా వాళ్ళు ఏమి కాదు, యూనివర్సిటీ డిసిజన్ మనం ఏం చేయలేము.
సబ్జెక్ట్ కొంచెం హార్డ్ ఉంది సార్ ఇది ఫస్ట్ ఇయర్ కంటే.
అవునవును ఫస్ట్ ఇయర్ చాలా ఈజీగా ఉంటుంది.
డయాగ్రామ్స్ తక్కువ ఉన్నాయి, మ్యాటర్ ఎక్కువ ఉంది ఇందులో కొంచెం.
అవును అందుకే
చాలా హార్డ్.
నేనేమంటున్నా అంటే షార్ట్ కట్ మీరు నోట్స్ తయారు చేసుకుంటే బాగుంటది అంటున్నా. నేను అదే చెప్తున్నా.
ఎస్ సార్.
షార్ట్ కట్ లా ఫ్లో చార్ట్ బస్. ఓకేనా?
Okay, sir.
ఓకే, థ్యాంక్ యూ.
Okay, thank you.
థ్యాంక్ యూ.